Welcome to today's guided notes on genetics. This presentation will walk you through the essential concepts of inheritance patterns, from basic pedigree analysis to complex statistical tools. You'll explore how traits pass from parents to offspring, why some inheritance doesn't follow Mendel's simple rules, and how scientists analyze genetic data. Follow along with your fill-in-the-blank notes, and check your understanding with the prompts provided throughout. A pedigree is a family tree diagram that tracks how a genetic trait passes through generations. Circles represent females, squares represent males, and shaded symbols show individuals expressing the trait. Generations are labeled with Roman numerals, and lines connect family members. Dominant traits do not skip generations, while X-linked traits may skip generations more often in males. When analyzing a pedigree, consider if the trait skips a generation, and identify X-linked patterns. Not all traits follow Mendel's simple dominant-recessive ratios. Incomplete dominance occurs when neither allele is fully dominant, creating a blended phenotype. Codominance shows both alleles equally, like blood types. Multiple alleles mean more than two options exist for a trait, increasing diversity. Understanding these patterns helps explain real-world genetic variation. Inheritance becomes complex when multiple genes interact. Epistasis occurs when one gene masks another, affecting traits like coat color in mice. Polygenic inheritance involves many genes contributing to one trait, such as skin color. This creates a bell-shaped distribution, showing a full range of heights rather than just tall or short. Consider why a mouse with black fur genes might still be white. Sex-linked genes are located on sex chromosomes, often affecting males more due to having only one X chromosome. Homozygous males express traits with just one copy of the gene. Common X-linked disorders include muscular dystrophy and color blindness. Understanding inheritance patterns helps predict trait transmission, especially when a male has a daughter with a non-carrier woman. Females have two X chromosomes, while males have one. Without correction, females would produce twice the gene product, causing imbalance. X inactivation forms bar bodies, silencing one X chromosome in each cell. This process ensures equal gene dosage between sexes. Calico cats demonstrate X inactivation, creating distinctive color patterns. Linked genes are located on the same chromosome and tend to be inherited together. Recombination frequency measures how often genes separate during meiosis, affecting genetic variation. Mapping distance helps scientists calculate how close genes are on a chromosome. The 50% rule indicates whether genes are linked or independently assorted. Mitochondrial DNA exists outside the nucleus, inherited from the mother. It affects energy production in cells. Chloroplast inheritance in plants involves genes responsible for photosynthesis. This type of inheritance is called non-nuclear, contributing to cytoplasmic traits during fertilization. Fill-in-the-blank checks help reinforce understanding. Chi-square test is a statistical tool that helps scientists determine if observed genetic data matches expected ratios. It answers whether any deviation is due to normal chance or indicates a different inheritance pattern. Calculating expected values and degrees of freedom helps make informed decisions about genetic hypotheses. Phenotypic plasticity describes how the same genotype can produce different phenotypes depending on environmental conditions. Temperature, soil pH, and UV exposure can alter gene expression, affecting traits like fur color in Himalayan rabbits or flower color in hydrangeas. Genes establish potential, but the environment determines how that potential is realized. Genetic disorders arise from gene mutations or chromosomal changes. Mutated alleles can affect protein production, leading to conditions like Tay-Sachs disease. Chromosomal changes, such as non-disjunction, can result in disorders like Down syndrome. Recognizing these differences helps explain the wide range of genetic conditions affecting humans. Congratulations on completing this genetics walkthrough. You've built a comprehensive understanding of how traits are inherited, from simple Mendelian patterns to complex genetic disorders. Review key concepts like types of dominance, polygenic traits, X-linked inheritance, linked genes, chi-square analysis, environmental influence, and genetic disorders. Use these insights to analyze complex inheritance patterns and understand how both genes and environment shape traits.